and the bony labyrinth of the inner ear. It is also going to help us understand a little bit more and know the structures, the allocation, specific cells of the sensory areas within the membranous labyrinth, as well as that of the semicircular canals and so forth, and also their different functions. And the last part, we'll be able to mention a few, mention the equilibrium pathways, as well as the hearing pathways, sorry. Okay. So just a little bit of revision from where we left off some 2 weeks ago. Our ear outline. So we have the conducting and the sensory structures. Our conducting structures, that's the outer ear, which comprises of the outer ear canal, the pinna, the middle ear comprising of the tympanic membrane and the ossicles, as well as the inner ear, which comprises of the semicircular canal. I apologize for that. Okay. Okay. So we say the outer, the middle, and the inner ear. Today we are focusing mostly, or we did the anatomy of all these. We are focusing mostly on the inner ear. So what are these? Our sensory structures, or the vestibular cochlear structures? We have the bony and the membranous labyrinth. If you remember what we talked about, the bony labyrinth, this is just the structures that are wrapped around in the petricious temporal bone. So we're talking about the semicircular canals, the cochlea itself, wrapped about around the modulus, which is the bony and the membranous labyrinth, the vestibular system, which is the auricle and the saccule. So for the bony and the membranous labyrinth, the easy way to remember this is that the bony contains the perilymph, and the membranous structures inside of these bony structures, they contain the endolymph. And if you remember, we mentioned that the perilymph is more of a composition same like that of extracellular fluid, which is CSF. That of the endolymph, it's filled or rich in potassium. And then for the vestibular system, we have the auricle and the saccule. So in this case, our auricle and the saccule is contained in the vestibule, which is our bony labyrinth. And then these auricle and saccule, they have the sensory organ specifically called the macula, which is contains hair cells and helps with all that we're going to discuss later. Also, we have the semicircular canals, their functional unit, which is a crista containing the cells we're going to discuss later. When you look at the auditory system, which is a cochlea, it's an organ of functional organ. It's an organ of Corti, which contains the hair cells that helps with hearing. So once again, vestibular system containing the auricle and the saccule, which are its membranous, this part. The vestibule itself is a bony structure. And then when it comes to the auditory, the cochlea is the bony labyrinth, and the organ of Corti, it's the membranous labyrinth. Why? Because it's the one that contains the fluid, which is the endolymph. Is somebody trying to call me?
Doc, I think there are some of our colleagues who are in the waiting room. I think today the weather is quite bad, the network, they are trekking out.
Oh, sorry. Let me see if I can add them back.
And Doc, if you may please make Andrew Piri the co-host, that way we don't have to disturb you every time.
Okay, so how do I do that? Me and Zoom man. Okay, so Andrew who? I put one more.
Andrew Piri.
Okay. Andrew Piri, okay. And then what do I ask?
Then you just go
Mm-hmm.
on the name.
Make host. Okay, I see.
Yes.
So I change. Okay. But will you still be able to share my screen? I mean, are you able to still see my screen?
Yes. So what I'll do, I'll just make you as co-host so that you'll be able to share your screen. I had made a mistake. Instead of making you the co-host, I made you a host.
Okay, thank you. I have just made you a host. Can you confirm, please?
Yes, yes.
Okay. So you can still see my screen?
Yes. Able
Okay.
to see your screen.
Yes, we can see your screen.
Thank you. So that was our outline. I know a lot of some of your colleagues or classmates came a bit late and missed out a little bit. So continuation is just the outer ear for conduction sec, receives sound waves. So it's shaped like a funnel, gets a sound wave, takes them to the middle ear. The function of the ear canal is to collect all these sound waves, project them on the tympanic membrane. So in the middle ear, which is the tympanic membrane, together with the ossicles, there's transmission from air, which is outside, into the ear canal, which is also air. And then they go to the fluid part, okay? To the fluid part. And remember that the fluid part, which is the middle ear, which is the inner ear, has to receive these sound signals after it passes through the bone, which is a temporal bone, from the bone now to the inner ear, and the inner ear is fluid. So from the eardrum, for it to be able to be projected properly into the inner ear, it has to be amplified about 22 times, okay, for it to be able to for the waves to travel through fluid. And how does that happen? The tympanic membrane is larger than the foot of the incus. Okay, so that on its own amplifies, helps to amplify the sound wave. Just a little reminder. Also, we have the inner ear, which vibrations transduced to the nerve impulses going to the acoustic nerve, which is what is our acoustic nerve? Can somebody help me? What do we call our acoustic nerve? Cranial nerve 8. Hello.
Vestibular cochlea.
Thank you. Okay, that means we are together. So vibrations transduced to the vestibular cochlea, then to the CNS, as you can see from our picture here. One structure I want you to take note of is the eustachian tube and the difference between an adult and a child's eustachian tube. What did we say last time? Can somebody help me? What was the difference? What is the difference between a eustachian tube or the auditory tube of a child and that of an adult?
We said in an adult, it is more slanty, and a child, it is rather horizontal.
Thank you. Another thing, yes, straight. Another reason that makes it susceptible to infections, recurrent infections of the ear, is that one is shorter, okay? First of all, the child is shorter and horizontal. An adult, it's slanted as well as long. Thank you. So for our inner ear, remember, this is inside the temporal bone, okay? So we have the vestibular. So vestibular is just a system with perception of body position. And then what are these function structures that helps us perceive? It's the utricle or utricle, as well as the saccule, and the semicircular canals. You have three semicircular canals. We have the anterior, we have the posterior, as well as the lateral semicircular canal. Also, it's also these structures are responsible for equilibrium, balance, okay? So for the hearing part, we have the structures we discussed. So here we are talking about vibrations at oval window, which are transduced to fluid. So changing of one form of energy into another. Sound waves into mechanical waves, then into nerve impulses, okay? So transduced to fluid, then the nervous energy to another when we say of transduction. So the two labyrinths that we have are the bony labyrinth. So these are encased within the preterist portion of the temporal bone. They're just inside the temporal bone. They consist of a vestibule. Okay. This is our vestibule here. The paper part where I'm pointing here, where the case, this is our vestibule. We have the cochlea here, and we have the semicircular canals. So it consists of the vestibule as well as the semicircular canals. And then for the membranous labyrinth, it's a sac within the bony labyrinth. So we have the macula as well as the ampullaris. So macula, ampullaris, if we talk of the cochlea, we are talking about organ of Corti. Is someone driving? Okay. So this is that the perilymph. Remember, we mentioned the main difference is that the endolymph is high in potassium, the perilymph is low in potassium, and the endolymph is these are produced by the stria vascularis, which is found in the organ of Corti in this case. Okay, so from all that we talked about so far, this is our sensory regions in the membranous labyrinth. When I say membranous, I mean inside the bony part, okay? So what am I talking about? I'm talking about organ of Corti, I'm talking about macula, I'm talking about ampullaris. So this is a vestibule. This is a saccule here, and then we have the utricle above it. And then these pink structures, these we call them on the semicircular canal, we call them the ampullaris or crista ampullaris. They are the bulging portion that comes and joins into the vestibule. So this whole structure here is a vestibule containing the saccule or the saccule and the utricle makes up the vestibule. Then we have the semicircular canals, but these bulging parts, just as we're entering the vestibule, is what we call the ampullaris or the crista of ampullaris. So each of these semicircular canal has its own ampullaris. Coming to the organ of Corti, our bony labyrinth, which is the cochlea, it has a membranous labyrinth called the organ of Corti. Okay. So on the hearing part, the functional unit of hearing in the inner cell, in the inner ear, excuse me, is a cochlea. Okay. So a cochlea consists of the vestibular membrane, the basilar membrane, the tight junctions, the stria vascularis, which produces the endolymph. We have the spiral ganglion, which is just cells below the organ of Corti. We have the tectorial membrane and the organ of Corti itself. So I want you to picture this. You put a markup on the table and let the markup lie not on its bottom, but on the side. It's like you spilled tea. Then you get a piece of paper, make a V, and insert that V into the cup. That will give you 3 portions divided by the paper V that you have made. So if we say this is our cup, and then you put, this is our paper here, this is our V, it will give you 3 portions. The first upper part, of course, here it's a cup. This will be, there will be a cup here. Picture this as the paper you've put in a cup facing you after it has fallen over. So the upper portion is what we call the scala vestibuli. Scala vestibuli, it's one portion. In the middle, we have this.
The DNA is quite.
Hello? Can you hear me?
Yes, Doc, we can.
Okay. So we have 3 portions after the V has divided. Okay. So we have the scala vestibuli. In the middle, we have a scala media, and then we have the scala tympani. So our Scala media is the point of interest. This is where you find the organ of Corti. Separating the Scala vestibuli to the Scala media, we have what we call the racina membrane. And then separating the organ, the Scala vestibuli and the Scala tympani, we have another membrane, which you call the basilar membrane here. This is the basilar membrane. This is a racina membrane, where my cursor is. Okay. So in this,
you need to increase the samples.
What samples are, can the one increasing the samples please mute or something? So our Scala media contains the organ of Corti, which is responsible for hearing. And then this is just a histological portion or part of how this looks like, the cochlea, okay? The Scala vestibuli, which is part of the bone, so it contains perilymph and the tympani. The Scala media contains endolymph, and Scala just means stairway. So here we have sound waves, which are converted into mechanical vibrations at the tympanic membrane, traveling through the perilymph, and then they come to the Scala vestibuli. From the Scala vestibuli, they make a U-turn. The point at which the Scala vestibuli and the Scala tympani come into contact is what we call the helicotrema. Okay.
Apologies,
so they
doctor,
down
for interruption.
Here.
Apologies, yes. Doc, for interruption.
No problem.
But
no problem.
I've got less than a minute. We'll just kindly ask if people can join using the same link.
Okay.
Yes, when it cuts.
Okay, so we restart. No problem. So, and at the end of a round window, at the end of the base of the cochlea, we have what we call the round window. We did all this anatomy, so I'm going to cruise through these. So, innervation of the cochlea. The cochlea is innervated by.
Oh, it has come.
Okay, so we're talking about the organ of Corti. This is what I was mentioning when I said, if you envision putting a paper, folded V paper into your cup that has fallen facing you, this is what you have. So this red line here is the racina membrane that separates the Scala media from the Scala vestibule. Then this other red line here below here is what we call the basilar membrane, separating the Scala media from the bottom Scala tympani. This is a cochlea when you unravel it. Okay. So you have the Scala media here. Inside the Scala media, you find the organ of Corti. And then we have the inner hair cells, which are the functional units for hearing, as well as equilibrium, balance. Above these cells, we have two types. We have the outer cells and the inner cells. So for the inner cells, they are a minority, only arranged in one row, okay? But the outer cells, they are a majority. They are arranged in many rows. They're about 90% of the hair cells are the outer cells. And then the inner cells are few, but 90% of your hearing comes from the inner cells. Okay. Sorry, just a minute. Forgive me, I'll mute you for a minute. My baby is crying. I need to give my.
Doc, if you can make me the host again.
Andrew P. How did I make you the host? I forgot.
Oh, you just go to my name on the list of participants where there's Andrew. Yes, then there are those three dots.
Okay.
There's more. Then I think on more somewhere.
Let me find Andrew. Andrew P. Okay. Then we go to more. Make host.
Yes.
Okay.
Okay, thank you so much.
I'll mute you for a minute.
Leo, explain to us about the organ of court, please, as we're waiting for the joke.
Apologies.
I have issues with my mic.
Okay, sorry. Okay. So we mentioned the major thing that you have to remember is that above the scala media, you are bordered by the retinar membrane. Below it's a basilar membrane. Now this basilar membrane is very important because its movement is what helps you hear. And then the inner hair cells, from the box, the inner hair cells, what am I doing? In the inner hair cells, here, this is where most 90% of your hearing comes from. The outer hair cells, they also participate, but that's less than 10%. Okay, so our organ of Corti has these two cells. Above this organ of Corti, remember that the hair cells, they have cilia. We have cilia almost everywhere, in the lungs, in the nostrils. So it's more like hair-like structures. The cilia from the hair cells, they are embedded in the tectorial membrane. So within the scala media, there is another membrane called the tectorial membrane. And then in this tectorial membrane, the cilia from the hair cells are embedded in. And below this, we have the basilar membrane. In the basilar membrane, that's why you find the dendrites of these hair cells, okay, and the cell bodies. This is where they're embedded. Cell bodies or cell bodies of the hair cells, they're in the basilar. The cilia is in the tectorial membrane. And then where does, which part of, which fluid is into this scala media? You have the indolent, full in potassium. So this is just what we were discussing. The organ of Corti lies in the basilar membrane of the cochlea. It buzzes in the indolent. The hair cells in here are the ones that are responsible for auditory transduction. Remember, when we say transduction, it's just one change of energy into another. The two types of the receptor cells, the inner and the outer. So the inner cells are few, arranged in only a single row, but the outer cells are in multiple, arranged in multiple rows. Why is this blue thing on this screen? So for the cochlear nerve, which is a cranial nerve 8, vestibular cochlea, it's the one responsible for innervating these cell bodies. Now, if you look at this, in this basilar membrane, you find that all these are fibers going to the cochlear nerve. They come in here and penetrate through the basilar membrane, innervating these whole cells, okay? And the impulses from here, they synapse, just here at the basilar membrane, and they go, the fibers forming the tract, that is the cochlea, that goes to join with the tract coming from the semicircular canal and the vestibule, which is a vestibular nerve. Together they form vestibulocochlear nerve. So what are some of the steps into this transduction we're talking about? So sound waves are directed from the tympanic membrane. When it vibrates, it causes the ossicles to vibrate, which ossicles are malleus, stapes, and incus. The incus foot plate is placed on the oval window. As it taps, the sound waves are being transmitted into the fluid inner ear, and it goes, when the sound goes there, it passes through the scalar vestibular first, and then meets in at the point where the vestibular together with the tympani, scalar tympani, where they meet the helicotrema. From there, passes through scalar medium. Scalar medium, the hair cells picks it up, and then it causes you to hear what's the generating process to go to the central nervous system. Okay, so this movement displaces the fluid in the cochlea, amplifying the sound. Remember, we mentioned how sound is amplified from the air field, which is outside, to the fluid field, which is a inner ear, which contains the receptors, our organ of Corti. So this is our more like a transduction. We've already discussed this structure on the left. Forgive me for this blue line here. I'll figure out a way of sending it. I will send the notes. Okay, so we have our organ of Corti here. We say it above it, where this cilia is embedded, it's tectorial membrane. Below it is a basilar membrane. They are innervated by the cochlear nerve, or they form tracts coming into formation of a cochlear nerve. Okay, so there are also some supporting cells here, other than the inner and the outer cells, which we are able to see in blue. So you find that these cilia, they have short and long ones. Someone is playing music in the background. That's nice music. Mute it for now. So we're talking about the transduction. So sound waves, which are transmitted via vibrations to the organ of Corti. So the auditory hair cells are mechanoreceptors. Okay, so just like they get this sound wave and turn it into mechanical as energy. That's what they mean. So which are located in the organ of Corti. The basilar membrane is more elastic than the tectorial membrane. This, our basilar membrane, is elastic, which helps or makes it able to move back and forth, like being pulled. It's more elastic than our tectorial membrane. So this causes the force, this helps with the force movement of the cilia as it pushes against the tectorial membrane. Okay, so bending of this cilia will produce a change in potassium conductance of the hair cells. So what they mean is, we know that the fluid in here in the scalar media, it's an indolent filled with potassium. Okay, so what will happen is, as sound comes here, it will cause vibration of the basilar membrane. As the basilar membrane is vibrating, it will cause movement of the cilia, okay, against the tectorial membrane. As it is brushing, this shear force, as it is brushing, it will cause, in between these cilia, you have ion-gated channels, okay, specifically potassium one, calcium ones. So these cilia in between, they have ion channels. And these ion channels, as the vibration causes the basilar membrane to vibrate, the sound as I'm talking, it causes the basilar membrane to vibrate. When the basilar membrane vibrates, it causes the cilia to brush on the tectorial membrane. When these cilia are brushing against here, the channel of the ion channel gated channels will open. So what will happen is that they only open when the force, the shearing force produced by the vibration of the basilar membrane is going from short cilia to longer cilia, okay? As it is ascending in cilia size, it will cause the channels to open. If it is vibrating and causing the shear force to come from the longer cilia to the shorter cilia, it will cause hyperpolarization. Why? Because the channels will close. They will not be able to open. Okay, so as let's imagine this is our longest cilia here on our right. So from the left, it's short. As the basilar membrane is vibrating, these channels will open. Why? Because we're coming from short to long. As they open, calcium and potassium, there will be influx into the cell because this fluid is filled with potassium. As calcium and potassium goes in there, it will cause depolarization, okay? It will cause a lot of positivity in there, leading to depolarization. When depolarization happens, remember that the calcium which is also going in there, other than depolarization, it will cause the vesicles in the cells to what? To burst, exocytosis. They will come to the base of the hair cell and cause exocytosis. What will happen is that they'll release neurotransmitters, specifically glutamate, and in some cases, aspartate. Okay, these neurotransmitters will fire. When they fire, they'll come to these neurons, neurofibers. When they fire, they'll send impulses into the auditory nerve fibers or the cochlear nerve fibers, which go to the CNS and causes you to have a response that someone has called out your name. Okay. So this is what these notes are just saying. You go through them as you read on your own. So therefore, oscillating depolarization, when we say oscillating, remember we said the basilar membrane is oscillating because of the vibrations that you're sending through the fluid as you speak. So it's moving back and forth. We call it oscillating. So the oscillating, depolarizing, and hyperpolarizing. Remember I said it depolarizes when it's coming from short cilia to longer cilia. And it causes hyperpolarization or switching off when it's coming from the longer cilia to the shorter cilia. Why? The channel gated are in between the cilia. So if it's coming, it's like it's closing them. So there is no entry of calcium, there's no entry of potassium, therefore there is no positivity in the cell, which results to it not being depolarized. And you won't hear anything at all. Okay, so what is this cochlear pathway? When they reach the CNS, the most important thing is depolarization and hyperpolarization in the transduction of sound. That's what I want you to focus on. Okay, so when the glutamate is released because of depolarization in these hair cells, it will go to these nerve fibers, turning these impulses into electrical impulses that will go to the central nervous system, okay? And then to the central nervous system, where will they go? These are the things I want you to just remember. Obviously, one is going to say describe. They want you to just know which areas. You have the cochlear nucleus, we have the superior ovillary nucleus in the central nervous system. We have the lateral lemniscus, inferior cochlearis, as well as the medial geniculate nucleus. That's where they will go. So this is just what, this is just a summary. It should have come after this. So information is transmitted from the hair cells of the organ of Corti to the afferent cochlear nerves. Remember, afferent nerves, they take something to somewhere. And then efferent, they take something from somewhere. The cochlear nerve synapses or neurons in the dorsal and ventral cochlear nuclei. Remember, we said it goes to the cochlear nuclei, it goes to the superior ovillary nuclei as well. So when they do that, if they take them to the dorsal and the ventral cochlear nuclei of the medulla, which sends out axons that ascends to the central nervous system. Some of these axons, they cross to the contralateral side. So you find that as I'm speaking from the left side, some of these nerve fibers, as they go to the central nervous system, they are going to go to the opposite side of the, so they're coming from the left. As they're going to the brain, to the central nervous system, they'll cross over to the right side, from the left to the right side. If they're coming from the left, some will cross over to the right side. That's what they mean. So we have the ones that cross and the ones that don't. Other axons remain espilateral. Espilateral just means straight up. They will remain straight up, and fibers from the nucleus of the inferior colliculus ascends to the medial geniculate nucleus of the thalamus. So we have about 4 of these where the fibers go. What's the point of this? I would have loved you to memorize this, but what is the point of this? The point is, if, for example, someone has a brain tumor on the right side, let's say on the right side of the lateral lemniscus, they will still, and then this completely damages that place, they'll still be able to hear some sound. Why? Because we have nerve fibers that sometimes cross, okay? They cross from the left to the right. So if it's the right side of a temporal region that is affected, they will still be able to hear some sound on the left side. Why? Because we have, on the damaged side from the left side. Why? Because we have nerve fibers crossing to the other side. Say a patient comes and then they have, let's say, a tumor of the ossicles, okay? Osteoma of the maybe incus on the left side that is causing the left ear not to hear clearly. If you look at them, examine properly, you're expecting them to completely have no hearing at all, but they'll be able to hear from the affected side because some of the nerve fibers, they cross over. That is the point of this, okay? So therefore, lesions of the cochlea of one ear will cause epilateral damage, deafness. So this is just, it will cause just one-sided. It's not like both sided will be affected. And then even that one-sided will not be complete, okay? They will still be able to hear some other sounds. That's what they mean. But I want you to pay attention on this and find your own time to go through your research and notes on this. But these are the 5 places that the cochlea are involved in the cochlear pathways. And then this is our summary, okay, of what we just discussed. Go through this yourself. We've already talked about it. We have sound waves coming in, reach the tympanic membrane, vibrates. When they vibrate, they get transmitted by the vibration of these ossicles by on the oval window. From the oval window, we have, so above here, what you're seeing, this is a scala tympani, where my cursor is. In between, scala vestibuli. In between scala media, where you're going to find the organ of Corti with hair cells. Which hair cells? Inner and outer. Which ones are more? The inner cells. And then below it is a tympani. From the tympani, scala tympani to the media, we have a membrane called the basilar membrane. Why is it important? It's important because it's elasticated and its movement causes the ion-gated channels to open, which causes sound transduction by potassium and calcium influx. When this happens, you have glutamate and aspartate released, which goes to the nervous system, central nervous system, which places? These places for you to perceive hearing, okay? This is just a summary of what we talked about. And when we come to the balance system, the vestibular system just means equilibrium, like maintaining the state of equilibrium. In this case, the way you're seated still. You are in equilibrium. When you move to look closer into your computer, what is causing you that to move? When you look to your left, someone is knocking, what is causing you that? What we want is we want to maintain state of equilibrium. We want to maintain balance. If you move, how are we going to help you maintain state of balance so you don't fall? Okay, so state of balance is maintained by detecting angular and linear accelerations of the head. When I say angular, I mean rotating, okay? You nod yes or no. Linear, I'm talking about you're driving, then you suddenly stop. When you stop suddenly, instead of braking slowly, your head will move back, then forward before it stops, okay? So that's what we're talking about, linear acceleration or horizontal in one direction. So the sensory information from the vestibular system, this is used to produce a stable visual image on the eye. So what we're talking about is the eye has to be able to see, oh, this one has moved. How can I help them stay still in that particular position? So we have the vestibular organ, which is located in the temporal bone adjacent to the cochlea. So we're talking about the semicircular canal and the vestibuli, which contains the utricle and the saccule. So the vestibular organ consists of a membranous labyrinth. Remember, when I say membranous labyrinth, I'm talking about, for example, in the cochlea, we have the membranous labyrinth in blue. Why this blue? Because it's the one that contains the organ of Corti. Okay, so any of the organs that contains the sensory function where the hair cells are, that's membranous. And it contains the endolymph, sorry. So the vestibular organ consists of the membranous labyrinth, which are the 3 perpendicular semicircular canals, the horizontal, the superior or posterior. And other names are anterior, posterior, and lateral. And the 2 otolith organs. Otolith, they just mean made of calcium and other components, as well as mucopolysaccharides organs, as we'll see later. So these are the utricle and the saccule in the vestibule. Okay, so now, why do they say perpendicular? If you look at the 3 semicircular canals, this is our image. The 3 semicircular canals, they are all arranged at 90-degree angle. These ones. If you are looking at them now, you will not see the 90-degree angle. Find some more images on the internet. They are all, and more info, advanced. It shows that they are all arranged at 90 degrees from each other, so that they are able to perceive movement changes in all directions. Okay, what are we talking about? If, for example, you look at it's not on this screen. If you look at the lateral semicircular canal, when I say no, okay, when I say I shake my head no, I'm using the lateral. When I nod my head yes, I'm using the anterior semicircular canal. When I move, like you're tired, you want to stretch your head, you move it round your shoulders, round your back like this. What I'm using is a posterior semicircular canal. That is what is indicated here, okay? And the semicircular canals are filled with endolymph, surrounded by perilymph, such as the cochlear we've talked about. So the semicircular canal are arranged at 90 degrees, like I said, to be able to detect motion in all axes. Each of these canals has endolymph inside them, okay, containing a large amount where they insert, okay? Where they insert, remember our pink image? Where they insert here, we have the ampulla here, crista ampullaris, where they insert, okay? This down here and this portion here, we have the utricle and the saccule. Here where they insert, we have the ampulla, crista ampullaris, or the ampulla. The ampulla is like the organ of Corti. It's a structure that contains hair cells. Moving on. So what happens is that, for example, and then in this, remember the organ of Corti, it has endolymph in there. But with the ampulla, crista, for the hair cells, they are swimming in what we call the cupula, okay? It's like a gel, it's a cupula. And then on top of the cupula, you have like stone-like structures that we call otoconia, otolith, and things like that. So in each of these canals, you have what we call the endolymph, and then this endolymph, gelatinous, it's gelatinous in nature. We call it the cupula. So just instead of how can I say, modified endolymph, but the composition is the same. The only difference is that one is a gel, another one is free flowing. It's a fluid. So for example, when you're moving in angular acceleration of the head, the cupula is displaced, causing excitation or inhibition of the hair cells. What do they mean? I'm driving, I want to start driving. Slowly, I've started my ignition. Then I quickly stop. My head moves forward and comes back. That is what they're talking about. I can either cause excitation or inhibition of the hair cells. Which hair cells? In the cupula. Which cupula? That found in the semicircular canals, okay? As well as in the utricle and vestibule, there are also hair cells. But the specific name for the portion in the hair cells, for the portion of hair cells found in the vestibule, we'll discuss later, we call it the macule, okay? So for example, counterclockwise, that is to the left, rotation of the head. When you rotate your head, you cause excitation of the left semicircular canals. But the right, at the same time you are doing that, will be inhibited so that your head does not get confused, like you have mashare or something, or you start nodding your head. So when you turn to your left, there's smooth movement, okay? Meaning the left side, the semicircular canals in your left ear, they've been excited. There's depolarization. So it will allow movement to the left. But at the same time, what happens? On your right side, there won't be any movement at all. You don't want to confuse your nervous system. It will cause hyperpolarization. The copula will not be moving. It will be still. Or if it does move, it will move in the negative direction. So that, which is the opposite direction of where the left was, you want to move in only one direction. So to make this simple, when you read a lot of literature, most people usually just get confused. Movement in one side of your body to the left, it will cause inhibition to the right. Okay, whatever is happening to say, yes, move, so you are stable. In the left, it will be the opposite in the right side. I hope it makes sense, but trust me, it will make sense when you read more notes. So in this, in the organ of Cort, remember we said we have hair cells with cilia, okay? We also have hair cells in the organ of, in the ampullaris. I couldn't find a better image of that. So when you look at images on the internet, they'll help you understand more, okay? I don't want to confuse you with things that you probably won't be able to use later on. But as a clinician, you'll be dealing with human beings, you need to have an idea of what we are talking about. That's why okay. So when we say the otolith organs, we mean we're still on the semicircular canal. So for the semicircular canal, I just want you to remember the copula as well as the knowing that one movement on one side causes negative movement on the other side, or causes opposite things on the other side. What things? Just movement of a copula will go in the different direction. So I was telling you that the hair that we have in the organ of Corti, we also have that hair in the semicircular canals, where in the ampulla, okay? So the ampulla contains the vestibular hair cells, which are embedded in the gelatinous mass. Which mass? The copula. So the vestibular hair cells, they differ from the cochlear hair cells, okay? Or the scala-media hair cells in the cochlea, in that the largest, the cilia, okay, so it's like the same cilia, except that we call the largest cilia the kinocilium, and the cluster of them, we call it the stereocilia. So look at it like stereocilia is short cilia, kinocilium or kinocilia is a large cilia. So afferent nerve fibers from the hair cells, remember this copula is moving. Let's say you are starting your car and then suddenly you stop, the copula is moving. When it moves, this mass, this gelatinous mass, when it moves, boom, what will it do? It will cause the hair cells also to move, okay? How will they move? The stereocilia will move towards the kinocilia, forming a shear. Okay, it's like a wind blowing in one direction. When this shear force happens and goes to the kinocilium, it will cause the ion-gated channels also to open. The same way, forming, causing potassium as well as calcium to enter. As a result, depolarizing the cell because there's increased positivity in the cell and sending a nerve impulse. It's the same concept of transduction. But For example, when the head is rotated counterclockwise, like I said, to the left, what will happen is this. The head is tilted to the left. The horizontal semicircular canal. Remember, I said the horizontal. When we say horizontal, we mean the lateral, okay? Canals, and they are attached to ampulla. They will rotate where? To the left. These are on the left side, okay? Initially, the cupula, which is anchored to the ampullaris. Ampullaris is where the fat part of the semicircular that goes into the vestibule. It will move before the indolence moves. So the cupula is just some indolence, but it covers these hair cells in the form of a gel. On the hair cells to also what? Be displaced, eventually causing rotation to continue. By the time the indolence comes to move again, to put your head back because you had put to the left, it wants you to put it in equilibrium. The indolence will also have to move. By the time it moves back, your movement, whatever you wanted to see on your left, has already occurred. And then what will happen is that if the stereocilia is bent towards the kinocilium, okay? So the hair cells in the ampullaris, you've moved to the left, and then these short cilia are bent to the long cilia of the semicircular canal. These are fancy names for them. What will happen is the cell will be depolarized and the firing rate will increase, causing the vestibular nerve to be excited and send signals to the brain. And then when you, that is on the left. On the right side, you don't want movement. So what is going to happen? It's the opposite. Which opposite? The cell will be hyperpolarized. So there is decreased firing of the afferent vestibular nerve. How? The kinocilium movement is from the kinocilium to the, which is longer, to the short, closing the gates. There's reduced firing action potentials. As a result, the right side won't be moving. It will stay still to maintain equilibrium, okay? So during the initial leftward rotation of the head, the left horizontal canal is excited and the right horizontal canal is inhibited. Remember, I told you that the lateral canals, they're responsible for your refusing, your no. The anterior canals are responsible for your accepting, you nodding when you nod your head. Yes, this movement is, which one is responsible? The anterior canal, semicircular canal. And then when you rotate your head back, forth, round in a circle, which one is responsible? The posterior canal. The second thing, when you are moving to say, I want to see what's on my left shoulder, counterclockwise. The kinocilium on the left side, the semicircular canal on the left side will cause action potentials to be produced, like the transduction we talked about. Why? Because there's movement from the short cilia, the stereocilia, to the long cilia, the kinocilium. And this force is being made by what? The cupula. So what we want is that this kinocilium is kind of longer compared to the otole. So it needs weight for it to be moved. Why do we have the cupula? That's its function. But the cupula on its own is not enough. On top of it, it has what we call the otoconia. That's why we're saying otolith organs. Otoconia is just some calcium stones with some micropolysaccharides. They will add weight so that they move this gelatinous cupula of the kinocilium and you're able to turn. But at the same time you're turning, the right side is not moving. What is happening on the right side? The lateral canal of the right side is depolarized. Why? You don't want to confuse movements by shaking your head like a mad person. The body is not created, God is not created like that. So what will happen is that there will be hyperpolarization here. What will happen is that there is no potassium from the rich indolence getting into the cell to excite it. And you'll be able to maintain your left movement. So whatever is happening on the left will happen on the opposite direction on the right. If it's on the right, it will be this direction on the left. Okay. Whatever is happening on the left, the opposite is happening on the right. How the movement of hair cells, it's the same, both in the semicircular canal and the cochlea. The only difference, things like cupula and the kinocilium and stereocilium. These are found in the semicircular canal. So the otolith organs, okay, we have the maculae. These are found in the utricle as well as the saccule. So whatever happens in the aortic on the sacral, it's the same thing, okay? So the hair cells in the macula, they're embedded in the otolith mass. We mentioned, we're talking about the copula. On top of it, we have what we call the otoconia, okay? This is what we just discussed. So whatever is happening on the right, it's the same thing that is happening on the left. So what are our pathways? So after all that happens, as I'm driving, then I suddenly stop, my head moves back, or a child is playing, they are rotating round and round. What is happening? Okay. One thing which is important is that the semicircular canal, they're responsible for what we call static equilibrium, turning to your left, nodding right and left. But the vestibule, where there's aortic and sacral, it is responsible for angular acceleration or angular movements. I'm talking about rotating movements, okay? So as all that is happening, the copula causes movements, potassium comes in, the whole story, yada, yada. What will happen is that nerves, the impulses they generate, they will either go to the cerebellum, the motor nuclei of the cranial nerve 3, 4, and 6, okay? Why this? We're talking about, when we say 6, we are talking about the vestibulo-ocular reflex. For example, you want to fall, then quickly you look down. What helps you to look? It's the eyes. You quickly look down, and the eyes, they see the cranial nerve 6 in. Oh, I didn't share, sorry. Okay. Can you see now? Okay. Okay, now I'm sure you're able to see. So I was saying the vestibular pathway. So all these afferent nerves, okay, from the vestibular hair cells, they terminate in the vestibular nuclei in the medulla. All the superior, medial, lateral, and inferior nucleus. Or in short, this you can go on your own, is that they go to these 4 major things. The cerebellum. Why the cerebellum? It goes to the muscles and instructs the muscles, okay? As well as maintaining equilibrium. So it goes to the cerebellum, it goes to the spinal cord, it goes to the eyes or the motor nuclei. So the motor nuclei of cranial nerve 3 and 4, as well as 6, especially 6, we call it the vestibulo-ocular reflex. Look at it. This is just instructing the eyes to see what you're supposed to do for you to be able to maintain balance. Of course, the eyes can't see. What we'll see is the tract that goes to the spinal cord, the vestibulospinal tract, via the vestibulospinal tract. So what will happen, for example, is you're walking, okay? Then suddenly you're driving and you suddenly stop, and then there's a person in front. You suddenly stop. You don't want to bash them. What makes you hand to stop or you to step on that brake? One, the tracts that come from vestibular or semicircular canal and that from the macule, they will cause your muscle to move. They go hand in hand. They synapse with that of the muscles and then causes them to move. Your hand will move on your steady, your foot will move on your accelerator or brakes, and you brake, you stop. If you are walking, you are spinning round and round. The macula in the sacral or utricle, they will sense, or this person, let's say, for example, you're jumping up and down, or you're in an elevator, it is moving up and down, up and down. What will help you sense that, oh, now I've reached, or now I need to stand properly so I don't fall, is those copula will move, send the signals to the vestibular nerve. The vestibular nerve with a tract that goes specifically to the spinal cord will tell you, oh, can you hold on to the door so you don't fall? You have to be in equilibrium. You hold on to the door and you stop the unbalancing that you are going through, or you stop the jumping up and down because maybe you wanted to miss your step and fall. So these are the 4 major places where these will go. They go hand in hand with other spinal, cerebral, as well as CNS impulses and innervations. Okay. If you can see, I've summarized this. I just want you to know these. You can understand them better as you read for yourselves, okay? Which nucleus and what, but all in all, they all end up to these four. Just like the cochlear ones. And the major things for the vestibule, the major thing we have in there, the sensory organs, we have the saccule and the utricle, and is that, okay, so semicircular canals responsible for static equilibrium, not moving the whole body, let's say, for example, just the head, okay? We said the lateral, the anterior and the posterior, which directions, I've mentioned it several times. And then where are these sensed in these pink positions I've mentioned here? These are crista ampullaris of the posterior, crista ampullaris of the lateral, crista ampullaris of the anterior. What do you find in here? Hair cells, okay? Specifically covered by the cupula, okay? And the otolith. And then you come to the vestibule. Vestibule, also part of equilibrium. What type of equilibrium? Dynamic. So we're talking about linear acceleration, deceleration. We're talking about rotation or angular rotation, okay? Which part is responsible? These two, saccule and utricle. But specifically, where do you find the hair cells in here? We find the hair cells in the macula, okay? And then in the cochlea, where do you find the hair cells? We find them in the organ of Corti, responsible for hearing. Okay, no one is going to ask you to describe, draw the tracks to the brain. All you need to know is the functional units, where the hair cells are found, and which places in the brain do they go to. Okay, and what else is more important? Yeah, those two. So remember, the vestibular nerve comes together with the cochlear nerve, and these two goes hand in hand. That's why you find that a patient comes in your clinic and they tell you, I'm feeling dizzy. And then this dizziness now has become worse. When I'm sleeping in my bed, I see the room moving and rotating. Okay. And how did it start? No, I faced my hearing was reducing. I can't even hear properly. I feel dizzy. Sometimes I vomit. It means that these areas are affected. They go hand in hand. The cochlea goes hand in hand with the vestibular to form the cochlear vestibular nerve. So you find that symptoms will be presented together. There'll be reduced hearing, poor hearing, there'll be loss of balance, meaning the semicircular canal or the vestibule in here. One of them has a problem, okay? What do you want to do? We call it the BPPV vertigo. Or if you want, just go and read on vertigo. It will help you a little bit, understand a little bit more of what we mean when the vestibular system is affected, the cochlea also is affected. When the cochlea is affected, same applies to the vestibular. You have some symptoms. Another thing you need to know is that when it comes to hearing, tracts that goes from one side of the brain, okay, so those that goes are sent to the brain is bilaterally. There are also others that goes and crosses to the other side from left to right. That's why you find that what you can hear on the left, you can also hear on the right. And when one person is affected, one side is affected, the other side will be functioning properly, and a little bit of function will remain on the affected side. Why? Because of the crossing tracts. Okay, and the basilar oscillating is very important. Structures and functions, what helps the basilar membrane to function the way it does. What are the special properties? It will help you, it will help in your clinical practice if you can focus on that. Okay, so we've come to the end of our balance and hearing system. Remember, cochlea is hearing, semicircular canal and vestibule is balancing, equilibrium balancing. What type of balancing? Semicircular canals are responsible for static equilibrium. And then the vestibule, which is the utricle and saccule, they're responsible for dynamic equilibrium. When I say dynamic, I mean movement, okay? Linear acceleration, angular acceleration, things like that. Okay, are there any questions so far? Are we together? Hello? Hello? Okay. I know you have other things and it was long. Yes. Okay, so when the vestibular pathway, there is interference on the vestibular. Remember, we said it's for equilibrium. So when there is interference of vestibular pathway, it will depend. Okay, say for example, I'll pick the one which is common and relatable to us, the motor nuclei of the cranial nerve 6. Okay, the ocular, the one responsible for the eyes. So if you say somebody punched or kicked or stabbed someone in the eye, so one, just that on its own, it will affect the eye, it will also affect the balancing. Why is it going to be affecting the balancing? The person may want to fall on the right side where the eye is affected. They cannot see properly on the right side. Okay, let's say it's in the dark, they can't see properly on the right side, or they want to get out of the bed in the hospital, they can't see properly, they're trying to balance on the right. It's going to be very difficult for them because they will not be able to perceive that disturbance in the equilibrium for them to balance properly. Okay, in that aspect on the right side. Why? Because nerve specifically going from that right ear or right vestibule to go and form what we call the vestibular ocular tract, they are affected. Another example, let's say the nerves that go to the spinal cord. You are drunk, you're staggering with balance. Those of us who drink, struggling with balance and you want to fall. Okay, there's a ditch on your left. Of course, your eyes are working, so the eyes will see at no, this side you're about to fall. But let's say maybe your leg is hurting, or you're wearing heels for girls. You're wearing heels and you can't, they are painful and things like that. You can't move properly. Already there is a defect with movement. So it is going to be very hard for you because you are drunk, the CNS on that particular side is also affected because of alcohol. For you to be able to avoid that obstacle on your left, let's say it's a hole or God, for you to avoid that obstacle, your eyes have seen, meaning your vestibular ocular is working well, and then the tracks going to the spinal cord, because of your heels, probably you will not be able to walk properly. So you have two obstacles in this case. And for you to balance, you find that maybe you try to avoid that hole and you end up falling. Why? Because tracts in the sinus, in the CNS, are also affected because of your alcohol. So you have three things affecting the vestibular cochlear tract. Why? One in the CNS, you wearing your heels and being drunk. Okay. So what will happen is that if it is hearing in the cochlea being affected, you have a chance of having some hearing in one ear because there are tracts that are crossing. But for the vestibule, it's very difficult. And most patients, they will present with vertigo, okay? Movement of things, or feel like they are spinning when they are not. Why? Because the cupula is not working well. Why? The hair cells are not being innervated properly. As a result, they're firing left and right, okay? Because we said that with the vestibule, the hair cells, when on the left, there is depolarization in that hair cell. Definitely on the right, there has to be inhibition so that you don't get confused. But when there's a lesion in this vestibular system, you find that there'll be overfiring anyhow, depolarization both left and right. And what do you think will happen? You have nodding of the head left and right like a mad person. Okay? Are we together? Okay. Any other question? One more before we go. I know you have class and it's long lectures are boring. Thank you. So this is just a tip of an iceberg. Read more on your own. And then where you find that you don't understand properly, there are a lot of videos all over. I like, anyway, you guys probably can't access it, but you can go on all these social platforms, look at the videos to understand better. One thing I want you to know, no one is going to tell you draw the pathways, okay? Just know where they go and what they affect. Thank you so much. Yes, sir. On hearing and balance, I would like you to broaden up a bit for your own understanding as a clinician. But for your exams and tests, these are enough. Okay, these are enough. I picked out a little bit of things for the test on the hearing part, but I want you to focus more on the things I've said, especially repetitively. Okay, so on the structure I've mentioned. On the hearing, I want the places where these go, like the last slide which you are seeing here. I want the sensory organs, which sensory organ of what part. Okay, and on transduction, it's very, very important. Okay, potassium is so important. Transduction in hearing, okay, and transduction in taste, transduction in smell. Those are the things I want you to focus on, because when you understand what happens for you to smell, to hear, you'll be able to understand what happens when someone is not able to do those things, and you'll be able to help them. Are we together? I'm not going to ask you to draw anything. Draw tracks to the CNS. It won't help you or me. Are we together? So for your test, focus on the things I've said repetitively. Those of you that have been attending even the previous ones. And don't worry, they're very easy. ENT, we don't like to make people's lives difficult. We just want them to understand, because trust you me, without your ear, without smelling, life is very bad. Trust me, it's very bad. Thank you so much. I wish you the best. How many more weeks before your exams? 2? Okay, all the best. But on the systems, I want you to broaden your reading, okay? The past lectures we've had, you don't need any of us. Those were enough. Thank you so much. Wish you the best. Bye.